Good afternoon, everyone. Thank you for joining us for the Kirby seminar today. My name is Nicodemus Sedla. I am chairing this seminar, but let me start by acknowledging the traditional custodians of the land which we gather today and pay my respect to their elders past, present, and emerging. I am talking from the land of the Gijikal people of the Eora Nation. And I also extend my respect to Aboriginal and Torres Strait Islander people joining us today. There are some housekeeping stuff that I need to tell you. The format of this seminar is that we'll start by presentation by Anurag Adhikari, who's my PhD student, and followed by Q&A. And then you can ask questions on the Q&A either throughout the talk or after the talk, and then I will try to answer the questions, or at least Anurag will answer your questions. So with that let me introduce you Anurag. Anurag Adhikari is a PhD, assigned a PhD student supervised by myself, Professor Rowena Bull, and Professor Andrew Lloyd. And his work involves on the nexus between the innate and adaptive in viral diseases, particularly in hepatitis C and SARS-CoV-2. So today we'll be talking to us about the longitudinal studies being doing on the role of the Fc receptor mediated phagocytosis in SARS-CoV-2 pathogenesis. Please welcome Anurag, and I'll get him to start the seminar. Thanks, Anurag.
Thank you, Nico, for this wonderful introduction. Before I start, I will like to acknowledge my respect to the Bedigal people who are the traditional custodians of the land on which this meeting takes place, and to elders past and present. Okay. So today I'm presenting a part of my PhD work, which is to understand the immune response, especially the COVID, the innate and humoral immune response after COVID infection, after COVID, basically, after SARS-CoV-2 infection. So this study is a cross-sectional and a follow-up study, which goes up to a year, and we follow the patients to understand the induction and maintenance of humoral immunity in this context. Currently, I'm a PhD candidate at the Viral Immunology Systems Programs at the Kirby Institute, University of Sydney. I'll start the presentation. Just give me a sec. Okay. I'll give you all a brief overview of SARS-CoV-2 infection and its pathogenesis, not in detail, but just a surface information for this presentation. So basically, I'll classify in a broad term protective versus non-protective immune response in SARS-CoV-2 infection. So I'm showing the airway epitheliums, which is the primary infection initiation site, where in dysfunctional immune response, you will see excessive infiltration of monocytes and macrophages and T cells, which would in turn induce lots of different cytokine storm, which in turn gives pneumonia, starts the pneumonia, and then of course, which is followed by pulmonary edema and basically destruction of tissue cells, and which in turn favors widespread inflammation and leading to multi-organ damage, which is a bad immune response, I would say, which of course you will see mostly in moderate to severely diseased individual during COVID. My interest would be more of a nexus, as Nicodemus told, between innate immune response, starting from the primary phagocytic cells like monocytes, neutrophils, and more of a antibody response. What kind of antibody are there during infection, and how do they orchestrate this immune cascade during early in the infection? That's my interest, and that's the gap as well in the literature at this point. And of course, in the protective response, you would see a better response in every aspect of innate and adaptive immune system. For example, you'll have a better neutralizing antibodies present in the system to neutralize the virus. The immune complex are efficiently cleared by the monocytes, which are one of the primary phagocytes which are employed during early infection. And with this good innate immune response, you will have a better adaptive immune response as well to follow, which would sustain the clearance and also maintain this a better adaptive immune response to a longer period of time, which is, of course, the infected cells are cleared better, the neutralization happens strongly, and then there would be a minimal inflammation and tissue damage or lung damage, I would say. So this three red part would be my focus, and especially focus on antibody-mediated response, especially the antibody response which initiate from the FC portion, which actually binds to these innate immune cells and helps clear the infected cells, virus immune complex, and other debris in the system. So moving on. Why this antibody response is interesting or you wanna work on, because there are really nice evidence at this point in SARS that if you do not have, or if you remove these FC binding region, which are the region responsible for binding to innate immune cells like monocytes and neutrophils, so you will have a systemic inflammation. And also in vivo model that has been shown that the viral burden increases compared to intact effector functions, antibodies. And it has been also shown that in prophylaxis or therapy session with other in vivo models, that if you can engage Fc gamma receptors, which are the receptors on innate cells, you will have less virus dissemination, decreased viral load, and of course decreased systemic inflammation as well. But if you compromise this Fc binding region of these antibodies, you'll have the clearance of basically the infection is delayed, which of course catches up with severity of the disease, which you wouldn't would be an unfavorable condition. And most importantly, recently has been shown in macaque model that with the vaccination, those vaccination where you induce antibodies which can have other than neutralizing function can actually prevent transmission, which is really important at this point, because we have like more than a dozen of vaccine at this point for human use, but almost none of them prevent the disease transmission, which is resulting in prolonged pandemic, as we see. So if we could work on transmission blockage as well, and then have those kind of vaccine that are really effective in reducing transmission, with the help of these kind of antibody, which have intact effector functions, that would be amazing. And that's the work I'm trying to do. I'm trying to develop evidence for what kind of Fc effector function are there in COVID patients, and how do they maintain over a longer period of after infection, and do they have any relation with as a protective immunity? That's the point I'm trying to figure out. So I do have few research questions which I will go through one by one. Before going there, I will start with the cohort we had that I worked on. It's a co-scene cohort led by Dr. Bull and Dr. Martinello, coronavirus outbreak samples in New South Wales. So that's the sampling study that I'm using for this study. So basically, the whole study design in a snapshot would be we would follow up. We'll enroll COVID-19 patients, get blood, and then from blood we'll use the plasma antibody for different assays, including the anti-spike IgG binding titer to understand what's the amount of antibody they have. And then we had developed the pseudovirus assay, which defines neutralizing function of this antibody that's presented on its plasma. And of course, this is another FC effector functions, which is a bit different than Phag effector functions that's called neutralisation. So that in this one, you will have monocytes, monocyte-based assay, which basically engulfs this immune complex, and then we measure the output, how much phagocytosis is being done. So for ease, I will be referring this antibody-dependent cellular phagocytosis as ADCP in the following slides. So basically, after in the first month, we had cross-sectional screening, and then we would, based on the data we got from all these different assays, we would follow them at 8 month, 4 month, and 4 month or 8 month, and then at a year after infection. So the first question we wanted to ask was, how does the severity of disease and patient's age plays a role in quality and quantity of antibodies that are produced after infection of SARS-CoV-2? And then more importantly, we wanted to ask if the antibodies are there, what kind of function they have. That was the primary question we had. So basically, in a nutshell, what we found was, as you can see in the graph, the green to red, different disease categories of COVID-19. And then you have the titer anti-spike IgG. As you can see, really high titer is being found in severe patients. Similarly, higher ADCP score. This is a P-score scoring system for ADCP. So you have higher ADCP among severe patients compared to asymptomatic. And the dotted line is healthy control and in each graphs. And you have, again, higher neutralization function in these severe patients. So it's very obvious that the more severe you have, better functions you might produce after infection. And also, again, we categorize our patients in different age group. As you can see, the older individual had higher titer, higher ADCP, and higher neutralization function. Statistically, ADCP was not significant among in between, but you can see the trend. So once we found that, we wanted to ask, how are these functions maintained longitudinally after the infection? So for that, we followed up the patient, of course. And then, as you can see in the first figure, there are 3 different groups of individuals. We followed them from 1 month, 4 month, 8 month, and 12 month. Dotted line is, again, the healthy control. As you can see, a significant drop in titer from first month to a year, which you would expect in any infection for the titers. And again, we analyzed the ADCP, which again you would see decrease in most of the individual, but some individual had a bit increase as well. We do not know the reason for that as well, but in general, as a trend, there is a decrement in as a mean count. Again, the same trend you would see in pseudovirus neutralization as well. So basically, the function seems to be going down, which you would expect, because if there is no infection, you wouldn't expect the immune system to sustain the immune response at a high alert level at all, because it's cost lots of energy. So again, when we saw the drop in functions, we wanted to ask the more interesting question again. So as you can see, the titer is decreasing substantially, even more than 90% from what we started with, and then the functions, neutralization functions and anti-spike P-score is also decreasing, but it's somehow maintained compared to the titer. But of course, you cannot compare in between the functions, but the extent of decrement is a bit relatively slow, I would say. And then we wanted to ask, okay, the titer are decreasing, the function are decreasing. What about the quality of antibody, which is a different thing than quantity? So for any biological function, you would understand the affinity against the immune epitopes or antigens would be crucial. So when we mapped the antibody affinity, we saw an opposite trend, where basically from 1 month to 12 month, the antibody affinity is increasing in plasma of these patients, which you would expect, because the longer the infection you have, So even though the titer is going down, there would be selection of better antibody producing B cells. And of course, you'll have a long lasting titer, which would be very small to measure with, but they will be of high affinity. And as you can see in the last figure as well, the fold change is tremendously high beside this patient, which we do not know why. So as a conclusion among these convalescents, what we found was there is a severity dependent and age dependent increase in functions, including titers, phagocytosis, ADCP, and neutralisation. And we also did a prediction methodology, where we put all the variables and try to figure out what variable would predict higher ADCP. We didn't do for other functions because ADCP was one of the primary output for us. And what we saw was if the patient have early on a higher anti-spike IgG1, you would expect the patient to induce or maintain higher ADCP in the long run. And then, of course, in longitudinal scenario, what we saw was although the titer or amount of antibody was decreasing, the affinity was increasing by many folds, which is a good news. And then what this part suggested to us, and then what we want to relay is, rather than having only the titer as an immune correlate or induction of immunity, we would suggest incorporation of at least neutralisation and ADCP function as a marker of immune response. So measuring antibody titers in sera might not be, optimal for knowing the immune response, and because that's what we saw in convalescent patient. And then as a follow up, what we wanted to ask is, we wanted to ask how early do these antibody are present in after infection in acute setting. So that's what we wanted to ask next. And then what we found was these anti-spike IgG, which are really high, have really high affinity, are detected as early as 10 days. As you can see, as a snapshot, this work was done in relatively low number of individuals because of the lack of the sample we had. But as you can see, as a snapshot of functions, if you see the first patients who was sampled within 10 days post-infection, post-symptom onset, you would notice that there is few functional readout, for example, for spike P score, but you'd notice a very, very low or almost negligible endpoint titer, which puzzled us in the sense that you would still have some function, ADCP, but you cannot measure, there is no detectable antibody, which was interesting for us. And then we wanted to ask, maybe the system we are using might not be sensitive enough to pick those low titer antibody. So what we did was we ran a SPR, which is a surface plasmon resonance. It's a very sensitive technique to measure the antibody or interaction between biological subunits. So what we found was really high affinity antibody that are present in a order of 10 power minus 12, which are really, really high. Even if you compare the monoclonal antibody that Donald Trump had, so after infection, these are of higher affinity than those as well, those monoclonal antibodies. So basically you have high affinity antibodies, but in very, very low amount, which still is giving some function. That's what we thought. And we wanted to see, because this is an early setting, you wouldn't only have antibodies, there will be other components in plasma as well, because these are native plasma that we use in the assay. So you will see in acute setting the inflammatory proteins, complements, and other opsonins that are actually responsible for inducing or assisting with phagocytic functions in early infection. So we wanted to see what's the role of these complements in this absorbed ADCP. So what we did is we set up a small, relatively simple experimental design to to block. Basically, what we did was we heat inactivated the plasma which would practically inactivate all the complement related proteins, and then you will only have antibody in there. So, and then the other assay, what we did was we actually blocked the Fc receptor, as you remember, the receptor that are responsible for binding the antibodies, which would engulf the immune complex. So we blocked the Fc receptors and then assess both of the settings to understand the role of complements and to understand the role of Fc receptor in this in the observed phagocytosis that's happening in early infection. So what we found was whenever we would hit inactivate the plasma, I will show you a snapshot of the same patient that we had, patient 1. You would decrease the ADCP, observed ADCP by more than 86%, 85%. But when you would block the Fc receptor on monocytes, which are used as assay cells, you would only decrease the ADCP, observed ADCP by 60%, which seems like there is the contribution of ADCP in early infection or early settings is a combination of both. There are the ADCP is happening through complements and Fc receptor in a synchronous manner, but we do not know what's the it's only a relative contribution at this point. We do not know exactly what and how much was contributed by each, but still this is a very interesting finding for us. So conclusion on early infection, what we could say is the ADC function, regardless of that's happening through complement or antibody, can be relatively observed early in the infection cascade, which is very important because that might have a role in driving the pathogenesis towards maybe protective or maybe detrimental. So more importantly, this hints toward a complex cycle between virus and innate immune system that needs to be understood better to have better therapeutic and preventive outcomes in future. So based on this whole scenario, this is more of a very preliminary data that I'm going to present you. So based on this, what we kind of propose is two different outcomes in made by antibody-mediated functions. For example, the first option would be, as you would see in dengue and RSV. What happens is there the non-neutralizing antibody early in infection or from previous infection would opsonize the virus, and then the monocytes or macrophages would take in the immune complex, which would result not in the sequestration or killing of this immune complex opsonized virus, but it will facilitate the production of new variants, and which would determine the disease outcome. And also in other respiratory infection, for example, of RSV, what happens is all these immune complex can actually infiltrate the whole the early systems, which would allow recruitment of these new immune cells, activation of complements, and of course would result in pro-inflammatory cytokine secretion from these monocytes and neutrophils, which you wouldn't want in a disease progression. So in this scenario, what we want to see is, because what we saw, the monocytes are capable of doing ADCP, could be from antibody, could be from complement. So we want to see in live virus, what role do these monocytes play if you would give them a live virus opsonized with antibody. That's the next step we want to take. It's an ongoing process, but I do have a very preliminary data that's very, very limiting, I would say. So the question was, if the ADCP is responsible for recruiting, to create a pro-inflammatory scenario early in infection, how do these antibody opsonized virus would be processed by macrophages, and what would be the immune response of these innate cells? That was the question we wanted to ask. And basically the idea was, the experiment design was, you'll have the antibody from patients, you'll opsonize the live virus, SARS-CoV-2, feed them to macrophages, monocytes, and then observe their cytokine response, and if they would produce any virions. It's basically a replication of the ADE phenomena that's happens. That's been shown in RSV and dengue virus infection. So for that, I do have a very limiting data that shows that co-incubation of SARS-CoV-2 with primary macrophage cells can produce pro-inflammatory cytokines. And as you can see in this figure, so there are few interesting cytokines that are IP10, IL-6, and IL-8 that are relatively expressed in higher than the control, which is virus only. So if you have virus only with macrophages, the response is milder. But if you opsonize with patient antibody, the response goes up. And particularly interesting are these cytokines, which has been shown in different study to be the hallmark or prognostic factor for severe disease induction. So that's an interesting thing what we're going through, and then we want to find out in more general trend that what kind of immune response the cytokine response these macrophages would give, and then if these immune response would be responsible for the disease manifestation. That's the question we want to figure out. So basically, what we want to do, we haven't done it yet, but what we want to do is update this model of SARS-CoV-2 pathogenesis, starting from airway epithelium, let's say. So basically, if you have, what we want to say is, if you have different antibody that are either neutralizing or non-neutralizing based on the response, the recruitment of innate cells could be different. And if that happens, and based on their immune profile, would this disease spectrum be different? That's what we want to ask. And then would the this pro-inflammatory monocyte macrophage axis by antibody and virus can change the fate? That's what we want to ask. And then, of course, what happens is once you have an antibody response after a vaccination or infection, do these antibody can mediate a protective versus pathogenic response? That's what we wanna ask. Yeah, as a conclusion, of course, this is the last one is limited by sample size. But what I would say is, for example, there are a few studies that I have cited in here have consistently shown these cytokines as a marker of disease severity. And then we want to ask, are these cytokines coming from the same mechanism that we are proposing, or of course could be a part of the mechanism that we are seeing? That's up to maybe future that we wanna figure out. But yeah, if that would happen, could better intervention for among those severe patients could be guided. And then, of course, the cytokine target for decreasing disease severity would be a interesting question to figure out. Yeah, saying that, I'd like to conclude the presentation. I'd like to acknowledge my supervisory team and, of course, the question cohort and entire Kirby laboratory team. And yeah, thank you. That's it. Any questions?
Thanks, Anurag. That was fantastic talk. And everyone, the question is open, the Q&A session. So if anyone had any question, could post a question, and then I'm not a Zoom type of person, but I think I might be able to work out. If the questions come, then I'll reiterate them. Any questions? The floor is open. Okay, while people are preparing to ask a question, I might ask you, Anurag, can you tell us you said that when you did multivariate analysis, can you tell us what multivariate analysis did and how do you rank them? And how did you end up saying that the anti-spike antibody titer was the highest, was the most significant predictor of phagocytosis?
Yeah, so it's a system based on multiple linear regression. So basically, you would rank you'll input all the variables that we have. For example, we start with age, sex, or gender. Then the titers for different subtype of antibody that are against the protein of interest, and other antibody functions. For example, what we did was we had titers, we had neutralization function, because we wanted to see, do these demographic and more of laboratory variables can predict the ADCP? That was the question. And then what we did was we modeled them in with linear regression to fit in, so that the idea would be, if you would enter each variable, what's its contribution to predicting or giving out the ADCP. And then based on that, you would say, for example, what we found was any individual who have anti-spike IgG1 at an instant would be predicting a higher chance of having ADCP as well. So basically, the weight the IgG1 carries, it precedes other variables, and then that's how you rank them. And then basically the model spits up the ranking system, and then based on that you'll say there's a good chance if you have anti-spike IgG in titer, you might have better ADCP, or if you have less IgG1 in the system, then you would expect less ADCP. That would be the idea.
Good. Any question? There's one comment from your supervisor, of course. Great talk, Anurag. Yeah, it's very good. That is, thank you, Professor Bull. Any question? Someone asked, for individuals whose antibodies underwent a decrease in affinity, do you have any working hypothesis for what could have potentially caused that?
That's a very complex question, and I don't have an answer for that. But what I would say, I mean, I couldn't say that. There will be multiple things. For example, you would start with the clinical condition they are, what kind of, if they have any genetic predisposition for antibody production or formation or maintenance, any ongoing co-infection, which our study doesn't account for. And, there could be myriad of possibilities that can relate the decreasing affinity. As you would see, I don't know if you're familiar with the measles virus, I think, if I'm sure. If you have measles virus or any other infection or co-infection, if you have measles virus, what it can do is it can actually decrease the antibody response or T-cell response to other infection as well. So basically, there will be unlimited reasons for that.
Can I just add to that question? It's actually an interesting question. Could they also have some sort of an autoantibodies, like an autoantibody that is actually to other proteins like interferons and everything that is diverting or a higher affinity B cells instead of having the functional like COVID, like the virus specific antibodies? Is that a possibility?
I would say, as I would know, there are many studies at this point where they have known and linked the presence of autoantibody with severe outcomes. But I actually haven't seen any study that would relate in between having autoantibody and reducing or impacting the actual antivirus antibody. But I don't know. Actually, I do not know if there would be any relation in between.
Okay. Any more questions? I just want to ask you about the increased affinity over time. Is there any evidence? What happens to the B cells? Are they undergoing somatic hypermutation, or are they changing? How does affinity mature and it becomes higher? And is there any evidence for that in other diseases or viral disease?
Oh yeah, that's a really nice question. So because our group is working on that, right? So what our group found in a nutshell, I would say that with time, there are selection of better affinity antibody producing B cells or memory B cells in a spectrum like post-infection. And then what our group has shown as well is the more severe infection you had during acute infection, and you are more likely to have to retain these B cells who might be producing better antibody in sense of affinity, because as the more mature or more selection of this B cell you have, there is a higher chance of having better antibody in sense of affinity. So I would say there are many evidence at this point among vaccinated individual, among infected individual, that based on vaccine as well, different vaccine have different response. But in a nutshell, you would say the more challenging infection you had early in the acute setting, the better and highly affinity selected B cells you will get in a long run, which would, I wouldn't say correlates directly, but which could produce better affinity antibody in the long run.
Okay.
But for us,
yeah.
Sorry.
There's another question. There's a good question. I think you've answered the question, but I think there's an interesting question here. It's actually not a simple question. You asked very, very nice question, wherever you are. If neutralizing antibodies and macrophages are associated with viral clearance, why would higher levels be associated with more severe disease? Is it because they are malfunctioning?
So to answer that question, I would say higher level. So you could have, I mean, there are lots of possibility, but in a dichotomous way, you could have better higher level antibody or non-better like autoimmune antibody that are less affinity or less specific to the antigen. So it depends upon what kind of antibody you have. For example, if you have really a high affinity, good antibody in higher level, it costs a lot of energy for the system to make and maintain those antibodies in the long run. So they wouldn't do it. But what you will see is low amount of antibody, circulating antibody in plasma, but who have better affinity in capturing the virus. That would be the ideal case.
But so, I think the questioner is asking a little bit more simpler. What they're saying is, if a patient has better phagocytosis, better neutralization, why do they end up having a more severe disease? They're like, is that how do you explain that? Because wouldn't they clear the virus?
That's the thing. Yeah, that's the thing. You will have to flip the study in a 180 degree to answer that. For our study, what it says is, once the patient have severe infection, the question is how they got onto the severe infection. We don't know. Our study doesn't qualify to answer that. What our study does is, once they have severe infection, what's the outcome of the other immune response, for example, immune response? So I cannot answer the question because our study is a bit different. We are not saying that they are severe because they have better antibody. No, we're saying the infection happened, they went to different disease state, and then outcome of that was antibody response that we measured. Maybe if they have reinfection tomorrow, if we could follow them up and they have reinfection and then the disease spectrum changes, then we could say, we could answer if patient having higher antibody, higher neutralization would have different disease outcome than they had in previous. That would be a really interesting question, but our study cannot say anything about that.
Or could it be simpler? Could it be simpler, like you've described to us, that people with severe disease have more opsonization that triggers more macrophage activation, more cytokine production, so they might look crooked, they might look sick. That means they're creating a lot of bystander effect. Whereas the mild ones, less antibody, less phagocytosis, they're actually like a piecemeal type of work they're doing. Whereas the severe ones, they are going full blown. I mean, if I was going to give an example, it's like, yeah, you have a target, a small target, but you blast it with a cruise missile. Means you will remove the target, but still you create a lot of bystander effect. Whereas if you use a sharpshooter, You will remove it, but you don't create a bystander. Maybe that's probably, that might explain it. I don't know, you are the expert, but I'm just proposing that possibility, because that's what you're looking at. If the opsonized antibodies are going to activate the macrophages to make a lot of cytokines that give grief to the patient, even though they're effective in taking up. Same thing with neutralization, I don't know, but it's a good question, I thought. Anyway,
Yes. Yeah.
any more questions? Any question? Any more question? I think, well, that's pretty good. We are finishing on time. It was absolutely fantastic talk. Anurag, nice graphics. And we do hope you answer the remaining of the questions that you have. And we can't wait to see. So please join me thanking Anurag. And I think we are all could go for early lunch. We've got 15 minutes to spare. Thank you
Thank you,
very
Nico.
much. Thank you, everyone.
Thank you, everyone.